we are getting into the topic, squalene hopene cyclase, a promising tool for biotransformation. And this is the topic which I have selected for presenting in this national webinar under the subject enzyme technology. Biotransformation is a very interesting topic in research. Um, you know, if you just make an assessment regarding the progress of research in industrial biotechnology, you can see the most vibrant field is a field where you can see the convergence of biology and chemistry. So biotransformation is an ideal example where you can see chemistry and biology being blended together. So it's very interesting to get into the various research activities in concern with biotransformation. And biotransformation, as we all know, it is concerned with the transformation of organic compounds into structurally related compounds. It may be with respect to the conversion of a toxic compound to a less toxic compound. It may be in connection with transformation of a compound into a more useful compound. So objectives can vary, but anyway, it is the transformation, structural transformation of an organic compound with the help of an organism or an enzyme isolated from the organism. But actual chemistry is involved in that. The reaction can be by hydroxylation, maybe by reduction or oxidation. It can be by isomerization, condensation, decarboxylation, dehalogenation. So any such chemical reactions can be there. We have plenty of enzymes from microbial sources capable of bringing either of these biochemical conversions. So, very fascinating field. Now, we have been doing research in this field for the last 22 years, or more than 22 years. And we feel that this research area is quite interesting because, as I said earlier, it involves biochemistry, microbiology, biotechnology, structural biology, some kind of chemical engineering, and whatnot. So you have a wide field, a very open field, and nothing can stop you. So you won't be stopped at any point because if one gate is closed, you have the other gates open. So very interesting field. And mostly, what is the biggest advantage of biotransformation is there is always a process simplification. If you just accept chemical methods to convert a compound from A to D, probably you have to convert A to B, B to C, and C to D. But if we adopt biological methods, if we adopt enzymes, you can most of the times convert A straight to D without B and C intermediate. So process simplification is the greatest advantage, and we have recognized that important advantage right in steroid biotransformations. So that's always been taken as a classical example to support the importance of biotransformation. And see, in most of the cases, we can see biotransformation is the only route for the production of or the conversion of a substrate into a product. And as I said earlier, there is reduction in the number of stages, there is process simplification. And more than that, all the biotransformation reactions are catalyzed by enzymes, therefore they are highly specific. Very specific reactions can be monitored, and it takes place at normal situations. Perfectly green protocol, and you can get sometimes novel conversions with enzymes particularly from microbial sources. In between that, let me ask you, I'm communicating with you, I'm audible, slides are visible, everything is okay? Are you following?
Yes, sir. Everything is okay.
Okay, fine, thank you. So that's it. And what I just going to present today, as I said, I'm going to talk about squalene hopene cyclases. And before squalene hopene cyclase an enzyme, before getting to the original research work, I just want to go through the basic concepts. I am very sure that each one present here is aware of the basic concepts, but I just want to revive it so that whatever I am going to discuss about our research is bringing some more clarity to you. as far as enzymes for biotransformation is concerned, these enzymes can be from bacterial species, it can be from fungi, it can be from actinomycetes. And when you try to do isolation of enzyme for biotransformation, the enzymes can be intracellular, it can be extracellular. If it is extracellular, things are very easy. But if it is intracellular, things are not going to be easy, because in most of the cases, if the enzyme is intracellular, the amount will be less and it will be inside the cell. Therefore, you are requested to break the cell. So there is an additional stage coming, additional methodology coming, and that can create so many challenges. So enzyme, and in our case, in the case of squalene hopene cyclase, it is intracellular. It is present only in less amount. So it can be inducible or constitutive. No, many enzymes are constitutive, it will be there always. But some of the enzymes are inducible. So they will be actually only in the presence of the substrate. Our enzyme is like that. And then in some of the cases, even if you have a good system for the isolation of enzyme, you need to apply methods of improvement. And when you are taking enzyme for biotransformation, you need large amount of enzyme. You cannot proceed with a small amount of enzyme. Therefore, you are requested to produce enzyme through fermentation. So bioprocess is to be exploited for large-scale production of the enzyme. And then once you have the enzyme, whether it is intracellular or extracellular, you need to purify the enzyme, because it's a chemical reaction, biochemical reaction. You are using enzyme as a catalyst, therefore enzyme should be absolutely pure. Otherwise, we won't be able to trace the fate of the substrate, the reaction involved in the conversion. So therefore, we need a purified enzyme. And as you all know, purification of enzyme is a challenging task. You have to apply several methods on a trial and error basis, and finally you have to make sure that your enzyme is pure. So we are going through, we are going through all these stages. When I present the research topic, exact research topic, we will be addressing all these issues. And then if you have enzyme with you, you need to worry about the structure of that enzyme also. What peculiarities there with enzyme? What all things are to be taken care when you apply enzyme for biotransformation. And quite a number of times, you need to overexpress it. And here, that particular situation is there. Squalene hopene cyclase is produced intracellularly. It is there along with the membrane, and it's available only in very less amount. So when you proceed towards utilization of squalene hopene cyclase for biotransformation applications, you need to get large amount. So you need to overexpress it. So you need to apply the techniques, basics of recombinant DNA technology, and you need to overexpress it. And we have done that. Again, when it comes to enzyme transformation, when you have enzyme with its own substrate, everything is fine. You'll be getting all the details of its analysis, optimal conditions from literature. But when you take an enzyme for a novel conversion, you have to standardize everything. Its pH, its temperature, its optimum concentration of the substrate, ionic concentration, everything needs to be standardized, which is a Herculean task. So we have addressed all these issues during our research work, and all the time we have to be very careful. You're dealing with a molecule which is having primary, secondary, tertiary, and quaternary structure levels. So if you want to exploit that enzyme, the activity of the enzyme, the native structure of the enzyme is to be maintained. So that's also very important. And we have faced so many challenges with squalene hopene cyclase in this context. And now coming into our work, the enzyme which we have been doing this work for the last 5 years. I'm going to present a summary of that work. And before getting into the actual research work, let me explain some of the details of this enzyme. Squalene hopene cyclase. This is an enzyme with immense potential, not addressed in a large way so far. Why I say it's very important. What is that enzyme? The enzyme is there in certain bacterial systems, and obviously it is capable of converting squalene to hopene. So that's it. What is squalene? Squalene is there as one of the intermediates in the terpenoid synthesis pathway. It is there in all the systems. So in bacterial systems, the squalene is getting converted to hopene. And that reaction is catalyzed by squalene hopene cyclase. So squalene hopene cyclase you find in microbial systems. Converting squalene to hopene. Hopene, what are hopenes? Hopenes are pentacyclic triterpenoid, and squalene is a linear compound. So that's the beauty of the reaction. Squalene is perfectly linear. Hopene is pentacyclic triterpenoid. And what is the function of hopene? Hopenes are having so many applications. Hopenes in the bacterial system, hopenes are contributing stability to the membrane. Otherwise, hopenes are having so many medical applications. It can be used in pharmaceutical industries. There are several other things concerned with hopene. So when it comes to SHC, I said the structure is very important. SHC is a homodimer with size ranging from 65 to 75 kilodalton. It's a monotopic membrane protein. I said it is a membrane protein. It's a monotopic membrane protein, means it crosses the membrane only once. You have the monotopic, ditopic, and oligotopic membrane proteins. Squalene hopene cyclase in bacteria is a monotopic membrane protein. The enzyme, the squalene hopene cyclases, they are having high chemo, regio, and stereo specific properties that can be exploited in chemical transformation. The highlight of this enzyme is this: very high chemo, regio, and stereo specific properties. We'll come to that, and I have to explain a little more. The enzyme is membrane bound and is present in only low amounts, and I have already mentioned that. That's a challenge. That was the biggest challenge which we were able to face. And then you have enzymes similar to SHC in eukaryotic system. It is there in plants, it is there in animals, it is there in fungi. And those enzymes you call as oxidosqualene cyclases. So squalene hopene cyclase in bacteria, similar enzymes in eukaryotic systems are oxidosqualene cyclases, because here the substrate is oxidosqualene. In bacteria the substrate is squalene, but in eukaryotic systems the substrate is oxidosqualene. So that's the difference. But anyway, this is in brief which I just want to tell you. So you have in prokaryotes conversion of squalene to hopene. Similar reaction, oxidosqualene is getting converted to cycloartenol. This takes place in plants, and you can see similar reaction in fungi and other species. We are concerned about only this. See, this is what I was telling. A perfectly linear molecule. Squalene is a linear molecule with unsaturated, and you can see getting converted to hopene, pentacyclic compound. So in a reaction, you can see five cyclization reactions are there. 5 rings are formed, and there are so many in a single step. In a single step, this enzyme is capable of introducing 5 rings, which you cannot find elsewhere. Not only that, I will come to this, and then you see this is the exact reaction. See, this is the steroid biosynthesis pathway, and you can see what is happening in fungi from squalene to tetrahydromanol, and you can see what is happening in plant, cycloartenol, oxidosqualene, squalene is converted to oxidosqualene, and then you have cycloartenol. And in animals, you have lanosterol, but here in bacterial systems, you have squalene hopene cyclase converting squalene to hopenoids. So we are worried about, or we are concerned about this, complex reaction. And I said, the enzyme is wonderful because it can even act in the absence of oxygen. It can act in the presence of oxygen, even the absence of oxygen. And I would say it is one of the most complex reactions already known. The most complex one-step biochemical process. Why it is like that? Just through a one-step reaction, the enzyme brings 5 rings. So this is the most striking element of this biochemical reaction. 5 rings, 9 stereo centers, and 13 covalent bonds in a single step, which you cannot find elsewhere in the subject of biochemistry. 5 rings, 9 stereocenters, and 13 covalent bonds. So therefore, that enzyme I said, wonderful molecule that enzyme is. And why it does all these things? What is the unique features of the? It is having a very unique motif. QW motif is a common. It's not so common, but it's a motif which you can find elsewhere also. It will find increased motif. And when we take different protein molecules, we work for we search for motifs. And SHC function, its a wonderful activity is somewhat linked to QW motifs, the number of QW motifs and the way in which these motifs are arranged. So when you get into the structure of that, QW motifs offer a significant role in this remarkable efficiency of squalene hopene cyclase, and it's a monotropic protein that I have already explained. So when you get to the chemistry, SSC mediated cyclization involves a series of discrete conformationally rigid partially cyclized carbocationic intermediates. Don't worry about the chemistry, but I just want to tell you the beauty of that reaction. That's why I have included this slide. No? It involves a cation formation by protonation of double bonds. So it's a very good subject for chemistry to get into. You can see reactive carbocationic intermediates on several occasions of this reaction. Intermediates, transition states are there. There is a ring closure is sequential. Beauty is there in the reaction. Rearrangement is there. Sequestration is there. There's a pi cation interaction. Cyclization through deprotonation. If you in one word, if you just say it is through deprotonation. So from chemistry point of view, from biochemistry point of view, it's very interesting to see this is the structure of the enzyme. As I have told you, it's a homodimer. Each subunit consists of alpha helical domains that build up a dumbbell-shaped structure. The first domain consists of a regular 6-barrel structure. You can see the structure of SIC. So we are talking about this protein that's very important. And thanks to these two great persons, how squalene hopene cyclase came into active research. That's also very important. So thanks to Dieter, thanks to Karl Perola of University of Tübingen, and Dieter is a German scientist. And these two persons were instrumental in bringing out the importance of squalene hopene cyclase, and they could publish few papers in European Journal of Biochemistry and Journal of Bacteriology. That happened in 1986, 1990, and 1992, only 30 years before. So these papers published by Dieter Ock and Karl Perola, introduced the squalene hopene cyclase to the research community, and thereafter many groups have been working on this SIC. So you can see almost all the works done in the last 30 years. The most important works done in the last 30 years. You can see large number of organisms being reported for production of SHC, for synthesis of SHC. And what can be the application? As I have already mentioned, it can be used for in lab conversions. You can use for the conversion of linear compounds to cyclic compound, and immense applications are there in pharmaceutical industry. Drug transformation can be effective. In fact, we have done some drug transformations. And hopenes are very important. Hopenes are having anti-inflammatory, anti-cancer, and antioxidant properties. So you can even use this enzyme for the synthesis of hopenes, large-scale synthesis of hopenes, because hopenes can even act as precursor for a large number of bioactive compounds. When you think of hopene formation inside the bacterial system, hopenes are formed in very less amount. So that's why, if you have enzyme, large amount of enzyme produced or purified, you can use that enzyme for in vitro conversion of squalene to hopene. That is being done. So you can have large amount of hopene, and that hopene can be used for the synthesis of large number of bioactive compounds. Can also be used in the pharmaceutical industry. So immense application is there for this enzyme. And I was justifying why we have started that work in our lab. Having fascinated by all these remarkable properties exhibited by squalene hopene cyclase, we started working on that. We started our work in 2016, and we were sure that the work is challenging. We were sure that we are going to face so many obstacles, but just we started that work. So I'm going to present the summary of works which we could do in the last 6 years, 5 to 6 years. And our object is very clear. We just want to work on SHC, squalene hopene cyclase. No, we don't want to purchase that enzyme, so nothing like that. No, we decided we have to get an organism producing SHC. That is very important. So we decided that, and then we thought of what can be the possibility of getting an organism. We made a hypothesis, and that worked well. That is the success of the entire approach, I would say. We just decided that we should go for endophytes associated with plants having high terpene content. Plants with high terpene content, obviously they will be having all different types of terpenoids. So nowadays we know that many of the properties exhibited by most of the plants are being done by endophytes associated with that. So with that basic knowledge, we thought of making an attempt to screen endophytes of various high terpene containing plants. We just started with more than 20 plants, in fact. I'm not going to show the result of all those things. Endophytes can be there in plant with high terpene content, contributing or regulating terpene biosynthesis. So this was the hypothesis on which we started the work, and fortunately, we could identify this plant, Murea koenig, and we should get more than 50 endophytes. We did many screening, primary screening, secondary screening, and all that. And finally, we got these 4 isolates as producers of SHC. So maybe you can summarize these experiments as a result of primary screening. We could get these organisms showing SHC synthesis. We then applied secondary screening method, and then we could see that Pseudomonas mendocina, MKE5, could produce a better amount of squalene hopene cyclase, and we identified this organism based on 16S rDNA sequences, and we could deposit that with that accession number also. So this is Pseudomonas mendocina. So we got that strain. Fortunately, our hypothesis was right. We could get a squalene hopene cyclase synthesizing organism as an endophyte of the plant Murea. So we started working on that, and this is the whole identity of the organism which we could the novel isolate, and how we could confirm the squalene hopene cyclase activity in this Pseudomonas mendocina. So we did HPLC analysis, and see the extract was taken, and that was mixed with the substrate. So you can see the HPLC profile of squalene alone. You can see the HPLC profile of bioconverted squalene treated with SHC extract from Pseudomonas mendocina. So we could very well detect the presence of squalene conversion. So hopenes were detected. Now, when you compare HPLC profile with the standard profile, we could see that, we could realize that hopenes were synthesized, and we just continued our experiment. But for better confirmation, we did GCMS analysis, and we got a very good peak, a unique peak in the case of SHC treated squalene. And when we did the mass analysis, it was very clear. The peak at 19.512 minutes that was resolved here, and the mass we got was exactly that of hopene. And even the mass fragmentation was perfectly with that of hopene. So we could confirm that Pseudomonas mendocina, the endophyte isolated from the plants Murea, could produce SHC. So that was proved beyond doubt. And then we proceeded towards isolation of the gene responsible for that. So primers were available. With that, we did this PCR amplification for all the 4 isolates, in fact. So very clearly we could get SHC gene amplified from Pseudomonas Mendocina. So that was again confirmed. We confirmed that with HPLC, we confirmed that with GCMS. We again confirmed that with PCR amplification, the same gene we could amplify it. And then we proceeded towards a homology modeling. I'm just presenting within half an hour or within 20 minutes our work, but this is a brief work, a brief presentation of 6 years work. So please consider that. And we just continued our homology modeling and docking and in silico characterization studies, and we just made the phylogenetic analysis of the SSC gene, and it was very clear that there was 100% similarity with that of the standard squalene hopene cyclase gene. And this is actually the homology model structure of squalene hopene cyclase, which we could isolate. And we did all validation studies, 3D structure analysis, Ramendran programs and everything. And we did the detailed docking studies using an ideal template, using a substrate, and we could use protein blast. We could use Swiss model. We could use parent tool and Ramendran analysis, and we could do all basic bioinformatics studies, and finally we could identify important amino acid sequences involved in the interaction between the enzyme and the substrate, as far as the in silico characterization is concerned. And so that was the second part. Second part is concerned with in silico characterization. I said in the beginning, the enzyme is produced only in very less amount. It is intracellular, it's produced in very less amount. Therefore, if we want to work with SSC, we need to overexpress the enzyme. So overexpression also we could perform. And this squalene hopene cyclase from Pseudomonas mendocina was introduced into the cloning vector PET28, and after the addition of cloning site for BAM-H1 and XH01, it was cloned into PET28A SHC. It was cloned into cloned and expressed in E. coli using PET28A, and it was overexpressed. And we can see this is actually the cloning of the SSC gene, and you can see the SSC gene being cloned, with all regulatory sequences in PET28. So we could do the transformation, and this is actually the E. coli transformed with the plasmid carrying the SSC gene, and we did the isolation of the plasmid, and we again finalized that SSC gene was cloned and overexpressed. And see, the enzyme produced was purified using histidine tagged SSC protein or nickel sapphirose column, affinity chromatography column, and that column, as you know, affinity column offers the highest level of purity. So we could get the overexpressed SSC at very high purity level, and that was confirmed with SDS page. So this overexpressed enzyme was purified, and its molecular weight was further confirmed with SDS page. So we could now say that we have enzyme, overexpressed enzyme. We have now enzyme in which we have already made in silico characterization. We have already purified that enzyme, so the enzyme is ready now. And this is almost a 4 years work, the result of a 4 year work, I would say. And then see, our objective was to exploit this enzyme for biotransformation reactions and see what we could do then. SIC is a wonderful enzyme. It can convert linear compound into cyclic compound, and it is an enzyme involved in steroid biosynthesis pathway. Thinking of that, we selected almost 8 to 9 drugs. Just a selection we made, an arbitrary selection we made, and then what we could do, the drugs which we could select: amlodipine, ethanol, clopidogrel, furosemide, glibenclamide, lisinopril, isradipine, metoprolol. So all these drugs are commonly available, and these drugs were selected. And you know what we did? We again did the in silico studies. You know, it's very difficult to find out in which case enzyme is acting in a better way. So we screened all these 8 compounds, and we did in silico studies. Now we just evaluated in which case the enzyme is acting in a better way. We analyzed the interaction between the enzyme and the substrate using in silico studies, and we could see that. So this is the details of the in vitro studies which we could conduct, and you can see glibenclamide. In the case of glibenclamide, it was very clear that the enzyme was interacting with that, and there is a chance of bioconversion. So the selection of the ideal targets for biotransformation was made based on in silico studies. That's again was a novel approach as far as the study was concerned. And as you know, when you proceed towards biotransformation, you know, the most important things which we have to do, you have to structurally analyze the product and the substrate using spectroscopical methods. So we just started doing this biotransformation tracing or profiling of biotransformation with FTIR, a Fourier transform infrared spectroscopy, and we could you can see the FTIR of the control here. This is the FTIR of the control sample with 10 millimolar concentration of glibenclamide at pH 6 in a reaction buffer. And this is very important. I was telling you this in the beginning. It is now very easy to say that 10 millimolar glibenclamide, pH 6, and reaction buffer and everything. But it was a difficult task to arrive at that situation. It was almost a 6 months work to standardize the ideal conditions for transformation. We just realized that there is a scope for glibenclamide to act as a substrate for this novel enzyme, but we were not able to, it was not there in the literature. For the first time, it was being proved. So now it's become our task to find out in what all situations the reaction is very effective. So that's what I'm telling you. In one word, I have just given the details of the enzyme reactions, but it took more than 4 months to standardize it. And now you can see, this is the control. Control in this means glibenclamide, which is not treated with squalene hopene cyclase, the FTIR of that. And here you can see the glibenclamide after conversion with purified SSC, the overexpressed SSC, the purified SSC that was used for the conversion of glibenclamide, and FTIR represented very clearly that glibenclamide undergoes structural changes. And as we know, FTIR gives a fingerprinting of functional groups. So you can see how these representations are different from the control. And not only that, we continued our experiment with LCMS this time. And LCMS also could give us, this is the LCMS of the glibenclamide taken. I mean, this is the LC. This is the LC of glibenclamide. I mean, not treated with the enzyme, the controlled glibenclamide. And you can see the peak. There is a peak at this peak. This represented glibenclamide, and this is the MS of that. This is the MS of glibenclamide, which is perfectly in agreement with the standard value. So it's obvious that your extract control is giving glibenclamide. And then see the difference. So this is the MS, this is the LC of controlled glibenclamide, and this is actually the LC of glibenclamide being treated with SHC. So we could identify a new peak here, which is not there here exactly. This new peak, there were so many other peaks. We resolved all those peaks, and then we could observe that this peak is very unique, and on mass analysis we could see that this peak represented hopene. So beyond now we could confirm it 1,000 times that this enzyme is capable of converting glibenclamide into a new compound, and this is for the first time a glibenclamide is tried, or any drug similar to glibenclamide is tried as a substrate for SSC. So that was a very good observation. And as you know, this is actually this is the conclusion. This was the conclusion out of that work. Glibenclamide is getting converted to its monooxygenated product. Actually, when you think of glibenclamide transformation in literature, you can see the first level conversion is by oxygenation. So this product, the molecular weight of this product could be established here, and now it's proved beyond doubt that SSC is able to transform glibenclamide into its monooxygenated product. Maybe that's the beginning. There is much scope for further investigation because once an oxygenated product from glibenclamide is formed, that can act as a substrate for several other compounds. We are in the process of tracing it out. We're just continuing that work. So this is all what I have to share with you. These are the final details of FTR and LCMS, and we could publish your work in several journals, the Biotech, a Springer journal, and in that journal we could publish in silico characterization and overexpression of scalene hopene cyclase from Pseudomonas mentosina. We could publish that in 2020, I think. And then we could publish this paper in molecular biotechnology, also published by Springer Nature. And you can see in vitro enzymatic conversion of glibenclamide using squalene hopene cyclase from Pseudomonas mentosina expressed in E. coli. The work which I have just presented got published in the end of 2020. And we could also get another paper. I haven't discussed that. Along with SSC studies, along with all those things, Pseudomonas mentosina was a very unique organism. We could also see the synthesis of a carotenoid, novel carotenoid pigment, and squalene hopene cyclase was contributing to that also. So I didn't mention that here because I just want to highlight only the transformation of drug transformation efficiency of squalene hopene cyclase. So we could also find out that these carotenoid pigment, this. So here you can see that in bacterial systems, squalene is not converted to oxidosqualene. You have squalene to hopenoids.
Okay.
But in plants, you can see squalene is converted to oxidosqualene, and then it is converted to cycloartenol. I just missed it. In plant, it is cycloartenol. In animal, it is lanosterol. These are all lanosterol, cycloartenol, and hopenoids. All these molecules are very important. They are having pharmaceutical activity.
Sir, this squalene has any connection with our vaccine preparation?
Sir,
which vaccine preparation?
No, sir. It is having connections. At present, we are exploring the possibilities of linking it to cholesterol synthesis. It is having connection with anti-cholesterol drugs. So we are in that line right now.
Is it available in this compound is in shark? Present in shark?
No. As such, it is not. As such, it is not there.
Okay.
As you are talking about hopenoids, sir?
No, squalene.
Squalene is available. You can get it from Sigma. Squalene is available. 2,3-oxidosqualene is also available.
Okay, thank you, sir.
Thank you very much, sir. Thank you, sir.